Hello, my name is Anders Matson. I am a fourth-year PhD student, and the title of my project is Enhancement of Natural Killer Cell Proliferation to Improve Cancer Cell Killing. Why does this research matter? Every year, 10 million people die from cancer-related deaths, so better therapies are needed to help treat those that have cancer. We propose that by focusing on NK cell immunotherapies, that we can improve the outcomes for those diagnosed with cancer. Some of the issues with current NK cell immunotherapies involve the persistence of the NK cells, or how long those cells survive, which isn't long enough to be used clinically. Second, the treatments themselves can be potentially cytotoxic, which leads to other health issues. Two things. What are NK cells and what are immunotherapies? NK cells are immune cells that specialize in targeting and killing virally infected cells or tumor cells. Immunotherapies are cancer treatments that use your own immune cells to target and kill tumors. A few types of immunotherapies that are currently being used in the clinics are cytokine-based infusions or the use of antibody therapies. So NK cells make perfect immune cells for immunotherapies because they are already great at killing tumors, so we are basically sending these cells to boot camp to become even better cancer killers. My research in the lab focuses on targeting the protease ADAM17 on natural killer cells. By targeting ADAM17 with an antibody that we call MEDI, we can enhance IL-15-based cytokine activation of NK cells, which leads to better proliferation of the NK cell both in vivo and in vitro. We are now studying the mechanism of how this enhanced proliferation occurs and the effects that it has on metabolism and effector functions such as tumor killing, with the goal of creating a better NK cell-based immunotherapies for cancer. Quickly, how do we conduct our studies? First, we collect blood and isolate the NK cells from the blood. Second, we culture the NK cells for 7 days with IL-15 plus or minus MEDI, which is the ADAM17 blocking antibody. And then third, after 7 days in culture, we collect the NK cells for characterizing and killing assays.
Going over some of our early data, on the left I have the data that shows the enhanced proliferation of NK cells when treated with IL-15 plus MEDI in vitro, and on the right I have the enhanced proliferation in vivo. Both in vitro and in vivo, you can see that when treated with IL-15 and MEDI, NK cells proliferate better than when they are treated with only IL-15. Next, these data show how our treatment improves tumor cell killing compared to cells that are only giving IL-15 therapies. The blue lines show NK cells only treated with IL-15. The red lines show NK cells treated with both IL-15 and MEDI. The darker red line shows that we can kill about 90% of the tumor cells in 2 hours when combined with a tumor targeting antibody, compared to only 75% killing when ADAM17 is not blocked on the cells, as shown by the darker blue line. How does this research impact NK cell immunotherapies? Our data shows that when combined with MEDI, IL-15-based activation has improved NK cell proliferation, persistence, and tumor cell killing. All of these lead to better cancer immunotherapy that can be used in the clinic soon to help reduce the number of cancer-related deaths each year. Thank you.